చిన్నగా. So welcome my dear friends. Madam, please click on slideshow. Today's our eminent resource person, Dr.P.Savitha Madam Garu, Assistant Professor, Head Department of Botany, GDC Luxetipet, Manchiryal District. Madam, please welcome and
Thank you,
start
sir.
your session, madam.
Okay, thank you,
Please
sir.
welcome.
So, good morning, everybody. This is my second class on Gymnosperms. So, if, up to yesterday, in our yesterday's class, we have learned about the morphological characters of the Gymnosperms. So, morphological and some anatomical characters also. Today, we are going to learn about the reproduction in Gymnosperms. So, many points are there which are helpful in competitive exams. So, listen properly and note the points. So, at last, after all classes, I'll be sharing you the multiple choice questions or bits regarding these Gymnosperms. Gymnosperms, let us start our class. When you come to the reproduction in Gymnosperms, these plants are either monoecious or dioecious. Monoecious means if the male and female reproductive parts are formed on the same
Madam,
plant
please click on
or
slideshow.
on different branches, that is called as monoecious. So, here the Gymnosperms are either monoecious or dioecious. Monoecious means bisexual. The male and female cones will be formed on the same plant, but they may be formed on different branches. Or dioecious means unisexual. Male plant will be different and female plant will be different. So in cycas, only in cycas we will be seeing the vegetative reproduction. In other plants like pinus and netum, we don't see the vegetative reproduction. Only in cycas we will be seeing the vegetative reproduction. That is by means of bulbils. These bulbils are formed at the basal part of the stem in the axils of the leaf. When these bulbils detach from the parent plant, they develop into independent plants. So in cycas, we'll be seeing the vegetative reproduction through bulbils formed at the base of the stem.
Excuse me,
Reproduction.
Madam, please click on slide. Slide is not visible. Only first slide is visible.
I'm
from the beginning.
Forwarding, sir.
No, madam. Slides are not movable. Please click on.
1 minute. I'll set it, sir.
From beginning. Slideshow from the beginning. Madam, left side. Left side from beginning.
Then I had to go again, sir. I had to go back again and I'll start again, sir.
Okay, sure. No, madam.
Are you able to see now, sir?
No. If possible, కింద బాటమ్ లో ఉంటుంది చూడండి మేడం, బాటమ్ దగ్గర కూడా స్లైడ్ షో ఇమేజ్.
ఇక్కడ ఏదో పైన ఏదో ఇది ఒక టేబుల్ లాంటిది వచ్చేసి సార్, అది కొత్తగా నిన్న రాలేదు, ఇవాళ వచ్చింది. అది మ్యూట్ స్టాప్ వీడియో.
మేడం, ప్లీజ్ ప్రొసీడ్.
Shall I start, sir?
Yes, madam, please proceed.
So, I'm Savitha from GDC Luxetipet, lecturer in Botany. This is my second class on Gymnosperms. In yesterday's class, I have taught you about the morphological characters, both and also anatomical characters. So today we are going to discuss about reproduction in Gymnosperms. Is it okay? Is it audible, sir?
it is audible. Please, you proceed.
Okay. When you come to the reproduction in gymnosperms, so we will get many points from the reproduction in gymnosperms for competitive purpose. When you come to the gymnosperms reproduction, they're either monoecious or dioecious. Monoecious means both the male and female cones will be formed on the same plant. They may be formed on different branches, but on the same plant. That is, they are bisexual. So monoecious means the plants here are bisexual, bearing both male and female cones on the same plant, but on different branches. When you come to pinus, we will be seeing the monoecious nature. And when you come to netum and cycas, they are dioecious, means male plant will be different and female plant will be different. So in cycas, we will be seeing the vegetative reproduction. Only in cycas, we will be seeing the vegetative reproduction, that is through bulbils. That is by bulbils. Bulbils, they form at the basal part of the stem in the axils of the leaf. When these bulbils detach from the parent plant, these bulbils, when they detach from the parent plant, they grow into independent cycas plants. So only in cycas we will be seeing the vegetative reproduction. In other gymnosperms, mostly we'll be seeing the sexual reproduction. So sexual reproduction is by formation of the cones. These cones are similar to the flowers in the angiosperms. Male cones are smaller and short-lived, and female cones are large, long-lived, and persistent. These female cones, they may be used for ornamental purpose, for decorating the houses in many places like Bengal, West Bengal, etc. Will be seeing these persistent cones. So pinus is monoecious. We have already discussed about the monoecious and dioecious nature. Next slide, sir. So in this figure, you will be seeing that the cones in cycas, cone in pinus, and male cone in netum. And this is the megasporophyll in cycas, and this is the female cone in netum. Here you will be seeing the ovules on the netum cone, female cone, and on the male cone of the netum, flowers are present, and microsporophylls in the cone of cycas, and megasporophylls and ovules on the megasporophylls of the cycas. This is about the images of these cones. So next slide. These cones are also called as strobili. Male cones bears microsporophylls arranged in spiral manner around the axis. Each microsporophyll has an expanded upper part ending in a sharp structure called as apophysis and a narrow basal part which is fertile in nature. In cycas, microsporophylls on its lower or abaxial surface, microsporangia are present in groups called as sori. In cycas, when you come to the microsporophyll, on the abaxial or lower surface, you will be seeing the microsporangia, which are arranged in a group of 2 to 5, which is called as sori. This group is called as sori. Whereas in pinus, only 2 microsporangia are formed on either side of the basal part of the microsporophyll, which appear as sac-like structures. In pinus, only 2 microsporangia are present on either side at the basal part, which appear sac-like structures. In microspores in pinus are yellow in color and with wings. In pinus, when you come to the microspore structure, they're yellow in color and they have wings which help in the dispersal process. So when the dispersal takes place in the pinus, the microspores, when they disperse from the pinus plant, we will be seeing as if showers of sulfur or yellow cloud is formed near the pinus plants. Hence, this is referred to as yellow clouds or showers of sulfur. Next slide, sir. So here in the diagram, we will be seeing the microsporophyll in cycas. Here you see at the tip a sharp curved structure which is called as apophysis, and at the basal part you will be seeing the basal part which is narrow in nature, and all over the lower part at the abaxial side you will be finding the microsporophylls. So this is the microsporophyll in pinus. These are the microsporangia where 2 are there on either side. on that side and this side, in front side and back side, you'll be seeing two microsporangia which form microspores or pollen grains. So next slide, sir. So male gametes in cycas are top shaped with 2 to 5 bands of cilia. This is the important character which we won't see in other plants. In cycas, the male gametes are top shaped with 2 to 5 bands of cilia which help them to reach the egg. So development of microsporangia is eusporangiate in gymnosperms. Eusporangiate means many initials are required for the formation of the microsporangia. Eusporangiate development means many initials form the microsporangia. Only one mother cell is not enough for the formation of the microsporangia. Many initials will take place in the development of the microsporangia. This is called as the eusporangiate development. Another type of development is called as leptosporangiate, where only one mother cell will be there, which forms the entire microsporangia. But in gymnosperms, we'll be seeing the eusporangiate development. Female cones bear megasporophylls, and in these cones, in cycas, cones are not formed. Compact cones are not formed. The megasporophylls are arranged loosely on the axis, and when you come to the pinus, compact female cones are formed. In cycas, megasporophylls consist of 3 parts: basal petiole-like structure, middle fertile region, and bearing ovules which may be 2 to 12 pairs in numbers, and upper part which is dissected pinnately and it is sterile. So megasporophylls in cycas, they are loosely arranged. They won't form any cone-shaped structure, and they consist of 3 parts: basal petiole-like structure, middle fertile region, bearing ovules 2 to 12 in number, and on either side of the petiole region, you will be finding in pairs the upper part which is sterile and it is dissected pinnately. And when you come to the needham megasporophyll, it bears 2 scales called as bract scale and ovuliferous scale. Bract scale and ovuliferous scale. Bract scale is present below the ovuliferous scale and protects the ovuliferous scale in younger stages. And after some time, it falls off. This bract scale, it falls off and it will be smaller than the ovuliferous scale in older stages. So the ovuliferous scale bears 2 ovules at its basal region. The ovuliferous scale bears 2 ovules at its basal region in needham. So next slide. Entirely different condition will be seen in the needham cone, where the male and female cones are formed on different plants, are divided into nodes and internodes. The cones of the needham are divided into male and female cones, both they are divided into nodes and internodes. At node of the male cone, scaly bracts, they fuse. Many scaly bracts will be there around the node, which forms a cup-like structure called as cupule. At the node of male cone of needham, many scaly bracts will be there, which fuse to form a cup-like structure called as cupule, bearing 3 to 6 rings of male flowers. Each cupule bears 3 to 6 rings of male flowers. Each ring consists of 25 to 30 male flowers. And these male flowers have a perianth. These male flowers bear a perianth and which have anthers at its apex. They won't have these petals and sepals. Just perianth will be there with 2 anthers at its apex. The cupule of the female cone bears 1 ring of female flowers having 4 to 10 ovules. This is about the cone of the cones, male and female cones of the needham. So next slide, sir. Ovules in gymnosperms are arthrotropous. Arthrotropous means here. Arthrotropous means here then chalaza, funicle and micropyle will be in a straight line. They are arranged on the same radii. So this type of ovules which won't have any bending or an angle, they are called as arthrotropous, which are straight in nature, which have the abundant nucellus, and only one integument is present, which is divided into 3 layers: outer and inner fleshy layers, which are parenchymatous in nature, and middle layer, which is stony in nature and made up of sclerenchyma. Outer and inner parenchymatous layers, which are fleshy in nature, and middle, which is stony in nature, and it is made up of sclerenchyma. If in Netum, when you come to the ovule of the Netum, an important character, a different character from the ovule of the ovules in the Gymnosperms is seen. A fan-shaped tissue called as pavement tissue is present at the basal part of the female gametophyte or embryo sac. In the ovule, embryo sac will be present beneath the embryo sac or the female gametophyte. You will be seeing the pavement tissue. In cycas, both siphonogamy and zoidogamy are seen. Means when you come to the cycas ovule, you will be finding a archegonial chamber, and when the pollen reach the micropylar region of the cycas, it produces a pollen tube which carry the male gametes into the archegonial chamber. And after releasing from the pollen tube, these gametes, they swim and reach the egg. This is called as zoidogamy. Here the gametes, they have motility and they reach the egg and fuse with the egg. That is called as zoidogamy. Siphonogamy means the pollen are taken by the pollen tube to the archegonial chamber. So this is called as siphonogamy. So this is about the cycas, and archegonia are present in pinus and cycas. Archegonia are present in pinus and cycas, but they are absent in Netum. You'll be seeing that in archegonia, the neck part and an egg venter is without walls, and egg will be present at the venter part, and neck will be there with the neck cells. 1 or 2 neck cells will be present, and venter part is with the egg where the male gametes will be fusing with the egg. In Netum, archegonia are absent. This is an angiospermic character, where in angiosperms, we won't see the formation of the archegonia. So hence, in yesterday's class itself, we have learned that this Netum is the bridge between the Gymnosperms and the angiosperms. So the cells in the embryo sac in the upper region acts as the egg cell and fuse with the male gametes in the Netum. Because of the absence of the archegonia, and the eggs will be present. So one of the egg cell in the embryo sac in the upper region, it acts as an egg cell and fuses with the male gamete. So next slide. So here in the diagram, you will be seeing the ovule of cycas. Here, micropylar region. From the micropyle, pollen tube is entering, where it is reaching to the archegonial chamber, and it takes the male gametes, where you will see 2 archegonia are present here with egg cells, and this male gametes, they fuse with the egg cell. Next is ovule of Netum. The basal portion, you are seeing the pavement tissue. See here, beneath the gametophyte, female gametophyte or the embryo sac, you are seeing a tissue which helps in holding the gametophyte at its position. These are about the ovules of the Netum and cycas and pinus. So next slide, sir. So endosperm in Gymnosperms is triploid. This is an important character which differentiate Gymnosperms from the angiosperms. Endosperm in Gymnosperms is triploid and formed before fertilization. Endosperm in Gymnosperms is triploid and formed before, it is not triploid, it is haploid. Sorry, here it is haploid. Endosperm in Gymnosperms is haploid and formed before fertilization. So monosporic type of embryo sac development is seen in Gymnosperms. Means a single megaspore is involved in the development of the Gymnosperms. And meroblastic embryo development is seen. That is, after the fusion of the male and female gamete to form the zygote, the lower part of the zygote develops into the embryo, which is called as meroblastic type of development. In pinus embryo, 16 cells are arranged in 4 tiers. First tier is called as open tier. Second tier is called as rosette tier. Third tier is called as suspensor tier. Fourth tier is called as embryo tier. So in pinus embryo, we will be seeing that 16 cells with the 4 tiers. In each tier, 4 cells will be arranged. So you will be seeing that first tier is the Open tier is called as open tier. Second tier is called as rosette tier. Third tier is called as suspensor tier, which helps in holding the embryo. And fourth tier is called as embryo tier, which helps in the formation of the embryo. The embryo splits and forms many embryos, which is called as cleavage polyembryony, which is common character in the gymnosperms. Cleavage polyembryony, it is a common character in the gymnosperms where embryo splits and forms into many embryos. So peculiar cells are seen in the development of embryo. In between the plumule and radicle, a structure called as feeder is seen in the younger stages in the neatum and helps in absorbing the nutrients. In neatum embryo, we will be finding that in between the plumule and radicle, a feeder is present in the younger stages and helps in absorbing the nutrients. So next slide. So this is about the reproduction in this gymnosperms. If anyone have any doubts, you can ask me. If anyone has any doubts, you can ask me.
Embryo is triploid, but endosperm is haploid.
Here in gymnosperms, I think in some disturbance, I have written as triploid. Endosperm in gymnosperms is haploid and formed before fertilization.
Yes. Regarding of
this you correct it, students. Endosperm in gymnosperm is haploid and formed before. This sentence you correct it. Any doubts regarding the reproduction in gymnosperms? So many points you will get from this reproduction part for making multiple choice questions. This part is very important in competitive purpose. So shall I repeat it once again? Stressing on the important parts. Sir?
Madam.
Shall I repeat some more points? Some of the points again?
Okay, madam, no issue. Whatever, if you are...
Students, did you understand this topic? Anyone is having any doubts?
Respond, my dear friends. Okay, madam. Economic importance is there, no?
You can ask me any doubt. If you want me to explain once more, I'll explain it again.
Okay, nobody can respond, madam. No issue. Economic importance is ready now, madam. Next class we will discuss.
Next class I will be explaining,
okay,
sir.
Madam. Okay, madam. Okay. Thank you, madam. Thank you very much.
Okay, okay, sir.
This is one of the important area.
Shall I stress on some important points in this topic?
Okay, madam. Please you proceed.
So share me the slides from beginning onwards, sir.
Okay,
so once again, I am repeating here. So main points. In cycas, we won't see the formation of the, in cycas, vegetative reproduction is by bulbils. Only in cycas, we will be seeing the vegetative reproduction, and cones are not formed, female cones are not formed in the cycas. And when you come to the cones, they are similar to the angiosperm flowers, and pinus is monoecious, whereas netum and cycas are dioecious. Monoecious means male and female cones are formed on the same plant. And when you come to the dioecious nature, male and female plants will be different. And so next. So change the slide, sir. Next slide. These images are okay, but... Sir, change to next slide, sir. So cones in gymnosperms are also called as strobili, where these male cones bear female, the microsporopils, which are arranged around an axis, each microsporophyll will have an upper part called as apophysis, which is curved in nature and which is sterile in nature. And microsporophyll at their abaxial side, they bear the microsporangia. In cycas, we see them in groups. 2 to 5 microsporangia are present in groups called as sori, whereas in Pinus, only 2 microsporangia are found on either side at the basal part. So these sporangia, in the microsporangia, microspores are produced after meiotic division. These microspores in Pinus, they are wing shaped, they have wings. And during the dispersal, you'll be seeing that because they are yellow in color, the yellow around the surroundings of where the Pinus plants are there, you will be finding that surroundings are yellow in color, which are called as showers of sulfur or yellow clouds. So this is about these are the diagrams of microsporophylls in cycas and Pinus. Next slide, sir. So male gametes in cycas, they are top shaped. This is the important character. Only in cycas we'll be seeing this. And 2 to 5 bands of cilia are present around the male gametes, which help them to reach the egg. Because of this only, zooidogamy is seen in cycas, and development of microsporangia is eusporangiate. Eusporangiate development is seen in this gymnosperms. Eusporangiate means many initials, parent initials are involved in the development of microsporangia. Female cones bear megasporophylls. The megasporophylls are different in different plants. In cycas, you will be seeing that they don't form any cone-like structure. They are arranged loosely around the axis, and each megasporophyll have 3 parts: basal, petiole-like structure or stalk-like structure, middle, fertile region, which have ovules in pairs. There may be 2 to 12 pairs of ovules present, and the upper part is dissected, pinnately dissected. So this is about the cone of the cycas. In Pinus, you will be seeing the megasporophylls are arranged compactly to form a cone-like structure, and in each megasporophyll, you will be finding two scales are present. 2 scales called as Bragg scale and ovuliferous scale. Bragg scale protect the ovuliferous scale in younger stages, and these ovuliferous scales, they bear 2 ovules at their basal region. So in Netum, these cones are different, entirely different types of cones are seen in the Netum. The male and female cones, they are divided into nodes and internodes. At each node of the male cone, you will be seeing scaly bracts are formed, which fuse to form a cup-like structure called as cupule. In Netum, cupule is seen. This cupule, it is formed by the fusion of the scaly bracts, and this cupule bears 3 to 6 rings of male flowers. These male flowers have a perianth, and perianth means a small. Perianth is nothing but a small structure which we can't differentiate between the petals and the sepals. Only a structure will be there. Each in this perianth, you will be finding that from the perianth, 2 anthers will be arising at their apex. The cupule of the female cone bears 1 ring of female flowers having 4 to 10 ovules. And ovules in next slide, sir. Ovules in gymnosperms are orthotropous. Orthotropous means they are straight in structure. Here funicle, chalaza, and micropyle, they are in a straight line. They are on the same radii. No bending is seen in the ovule. So different types of ovules are seen. We have learned different types of ovules. Orthotropous, anatropous, sursonotropous, like this. Many types of ovules are there depending upon the angle between the funicle and the body of the ovule. So in gymnosperms, no angle is seen. Straightly, this funicle, body of the ovule, and micropyle are arranged on the same radii. So with the abundant nucellus, this ovule is present, and only single integument is present around the ovule, which is divided into 3 layers or 3 envelopes are present around the ovule. Inner and outer are fleshy with parenchymatous tissue, and middle one is stony with sclerenchymatous tissue. In netum ovule, you will be seeing a different structure, which is called as pavement tissue at the basal part of the female gametophyte. After the formation of the female gametophyte, at the basal part, you will be seeing a tissue called as a pavement tissue, which helps in holding the female gametophyte in its position. In cycas, both siphonogamy and zoidogamy are seen. Siphonogamy means you will be seeing that the pollen tube is formed and it carries the male gametes to the pollen chamber or archegonial chamber. Zoidogamy means the gametes which are liberated into the archegonial chamber, they swim to the egg. This is called as zoidogamy. So in cycas only you will be seeing the siphonogamy and zoidogamy. And when you come to the pinus and cycas, archegonia are present. But in netum, you'll be seeing archegonia are absent. Archegonia in cycas and pinus, they have neck part and a venter which is without walls, and only eggs will be present. Egg cell will be present. And in netum, this archegonia is absent. Instead, you will be seeing that from embryo sac, from the upper part of the embryo sac, a cell will enlarge. One of the cell from the upper portion of the embryo sac, it enlarges and to form the and fuses and becomes the egg and fuses with the male gamete. So these are the diagrams, ovule of cycas and ovule. So endosperm in gymnosperms is haploid in nature and formed before in fertilization. And this is the differentiating character, which is a very important character regarding competitive examinations. It is asked many times. So endosperm in gymnosperms is haploid and formed before fertilization, whereas endosperm in angiosperms is triploid and formed after fertilization. So this is the differentiation character between the gymnosperms and the angiosperms. And when you come to the development of the embryo, monosporic embryo development is seen in the gymnosperms. Monosporic means only we will be seeing that the megaspore mother initial cell, it undergoes meiotic division to form 4 megaspores. Out of those 4 megaspores, 3 megaspores will be disintegrated. Only 1 megaspore will be present, which develops into the embryo sac. So only 1 megaspore is involved in the development of the embryo sac in gymnosperms. Whereas in angiosperms, we will be seeing many different types of embryo development will be seen. Bisporic will be there, tetrasporic will be there. In gymnosperms, only monosporic type of embryo sac development is seen. And the embryo development is meroblastic. Meroblastic means the lower part of the zygote develops into embryo. Only the lower part of the zygote develops into embryo. In pinus embryo, 16 cells are arranged in 4 tiers. The upper tier is called as open tier. Second tier is called as rosette tier. Third tier is called as suspensor tier. And fourth tier is called as the embryo tier. This embryo tier develops into embryo. The embryo splits and forms many embryos, which is called as cleavage polyembryony. This cleavage polyembryony, it is a common thing in the gymnosperms. Many embryos are seen. Only one embryo gets successful, but only one embryo develops successfully into a new plant. So peculiar cells are seen in the development of embryo in netum. In netum, a special character is seen, that are peculiar cells are seen, and also feeder is seen in between the plumule and radicle, which helps in absorbing the nutrients. So this embryo develops into later around the embryo, whatever ovule is present, it transforms into a seed, and these seeds, they develop into plants. So in cycas, the ovules are the largest in the plant kingdom. The seeds and ovules present in the cycas are the largest. This you have to keep in mind. This completes our reproduction in gymnosperms. Sir, okay na sir? Thank you for giving me this opportunity.